Hello, and welcome to the Biology 201 course overview for the section for Dr. Faison for fall 2016. Got a couple of quotes up here that I think are very important for you to have an understanding of what it is that we're doing here. So the first of those is there is no learning without practice. And what that is really about is the fact that you will have to do a substantial amount of work to make sure that you have learned and accomplished all the goals for the class. Second is value knowing. One of the things that are important in here is that you actually know the material. And what we mean by knowing is that you can, with books and notes closed, actually come back and talk about facts from this course that you actually know it. And finally, humans can do what humans have done. What that means is that it is very possible out of this class. Folks have done it in most all previous semesters, and that being the case, you can also do it because, again, humans can do what humans have done. Some quick notes on the course itself. Here's all of my information. It's actually found also on Blackboard in the course administrative section, and I'll go through the Blackboard section for the course shortly. My office for now is in 100M Owens Hall. My email address is mfaison@bsu.edu. The course runs apparently Tuesday, Thursday from 9:30 to 10:50. My office hours are Tuesday and Thursday from 11 to noon. So basically right after class. Other times are by appointment. You can use this number to call me or to text me if you have issues that need to be discussed. So the purpose of this discussion is to provide a sense of the organization of and the philosophy behind the course and the way that I actually deliver the course. This is not really a syllabus overview, even though I do talk about some specific sections, including how you get your grade. The syllabus is available on Blackboard, and you should download and read it. As with most of the courses, as you go through your sequence as a biology major, what courses do is stack and add new material on as you progress and gain more knowledge. So what we do here is we add additional depth to several concepts from Biology 120. It is, at the end of the day, your responsibility to ensure that you have your understanding of Biology 120 concepts. Once we move into that material, what is that you actually have the background and we're ready for the new material. Videos on Biology 120 concepts that are needed for each unit are on Blackboard, and we'll discuss the Blackboard for this class shortly. Look at the evolution from Biology 120 to Biology 201. What you see on this page is the major topics that you guys had in Biology 120. We can add to that then the topics on the right that you will now see in Biology 201. So what this is the major topics that you had in 120 that will be added onto in 201. A lot of it's the chemistry stuff, membranes, energy and metabolism, so some biochemistry, cellular respiration, cell communication, cell cycle. What we don't really do is genetics. Now what you see is color coding of how these actually map onto each other. So biological chemistry were your first couple of topics. We sort of flipped some things in terms of energy and kinetics, being your energy and metabolism from before, membranes and membrane transport. Energy metabolism really deals with your cellular respiration and photosynthesis, so we had additional depth on that. We had a lot more in terms of cell communication, cell cycle, cell death, meiosis, even though in this case, as you'll see, that we don't do as much talking about mitosis and meiosis specifically, and gene expression. Again, what we don't do is the genetics. There's a whole course that you take on genetics in and of itself. We add as we go from Biology 120 to 201. So what we're going to do in this top in the section on biological chemistry is we're going to re-emphasize self-assembly, as well as give some additional discussion of bonding and protein structure. For the energetics and kinetics calculations, what we're going to add on is actual calculations, particularly dealing with free energy as well as enzyme and receptor kinetics. For membrane transport, what we're actually going to do is apply some of the membrane transport principles. So the discussions of passive transport, active transport, etc., to actually understanding membrane potential. So that's electrical activity across the membrane, as well as have some discussion of vesicle budding and fusion. Energy and metabolism, again, we're going to go back to some of these energetics and membrane transport principles and now talk about those in the context of energy metabolism, in the context of cellular respiration and photosynthesis. Reception and signal transduction, we're going to actually learn and use specific signal transduction pathways. And so this is going to be a deal where you actually have to sit and now learn the specific pathways. Gene transcription, we will focus on transcription factor function, as well as the mechanisms for silencing eukaryotic gene expression and the effects of alternative splicing on eukaryotic gene expression. When we go into the cell cycle, we are actually going to focus on the regulation of the cell cycle. So it is going to be assumed that you know the steps in the cell cycle. And now we're going to focus on the signaling that's involved in that regulation, as well as for cell death. And then we're going to apply that knowledge related to signal transduction, gene expression, cell adhesion and motility, the cell cycle, and cell death to understanding tumorigenesis, which is the formation of tumors, and metastasis, which is the spread of tumors through the body. And so we're really going to do this in a lot of the cell biology that you learn throughout the semester in the context of cancer. So to get us going early, one of the things that we need to do is review some major points from Biology 201, those essentials being bonding, gene expression, and signal transduction. So what I have created for you is this review document of Biology 120 major concepts. So the major concepts are going to be bonding, gene expression, signal transduction, and we're going to start with two major types of signaling: G protein coupled receptor signaling and receptor tyrosine kinase signaling. And one of the things that this can also help to drive home the point of, I hope, is that it is presented in different ways across different textbooks. So this first column is the Freeman text, which was the course which was the book that you used for Biology 120. So this is where you can find the important topics for this review in your Freeman text, including the most important figures. Your current text is Becker, and so you can use this in the Becker text. There are also 3 really good cell biology texts available free online through the National Library of Medicine. And so what these are links to is where you can find this material and the most important figures in those textbooks. So then the key things that I want you to be able to do are listed here. So we want to be able to name and distinguish between the major types of bonds that are found in biological systems. We really want to have an understanding of what the term gene is and what gene expression is. We want to be able to list and describe the major stages in eukaryotic gene expression and the major forms of regulation of gene expression and which stages of gene expression that they actually regulate. And for the signal transduction component, the major forms of intracellular signaling, the major stages in a signal transduction cascade, and all of that part can be found. Transduction overview, differentiate between signaling through hydrophilic molecules and hydrophobic molecules, the steps involved in the deactivation and activation cycle of a G protein coupled receptor, and that'll be found in your G protein coupled receptor section, and the steps from ligand binding to activation of a ligand binding to a receptor tyrosine kinase to activation of RAS, which is the downstream target, and you'd find that in the receptor tyrosine kinase section. Now is give you, actually, let's talk about how your grade is going to come, then we'll move into Blackboard. So this is how your grading is going to come. 15% is homework, in-class activities, participation. 20% are in-class quizzes, 30% are online quizzes, 25% of your exams, and 10% will be a final project. So first, for your exams, they will be at the end of each unit, and a unit for us is basically 2 different topics. I'll go through the course schedule as well. Require Scantron, so on exam days, you should bring your own Scantron and a pencil to class. In-class quizzes will occur every day in class. They may be at the beginning or they may be at the end of each class period that's not an exam day. They will be 5 to 10 short answer questions, and they will be on your own paper. I'll also use the quizzes as a role. Online quizzes are available through each section, throughout each section of the course, which basically means from the time that we move into a new topic until the day that we finish it. And those online quizzes will close at noon on the last day of each unit. They are 10 multiple choice questions, which the computer will select from a pool of questions that I give it. They can be taken in a limited amount of times. The grade is the average of all attempts. Now, I'm going to caution you guys on these quizzes that some folks like to go in and just take them one time and be done with it. Learn from, which is why I have them up there so that you can take in a limited amount of times. A single shot 100 doesn't tell you much. It's when you can get multiple 100s on this quiz consecutively that tell you that you really know the material. And then if you did poorly the first time and you need to bring that up, you can keep taking the quiz in order to bring that grade up. You will have frequent homework assignments. Some of which will be peer-evaluated, some of which you will turn in, some of which you will do on mastering biology. I think I will say most of the mastering biology discussion for next Tuesday, but we can have a brief talk, actually for Thursday, but we can have I'll show you some things briefly with it. As far as your participation grade, it starts with 100, and you can chip away at it in turn by not participating in class. Oh, one note with respect to your in-class quizzes, there are frequent problem sets that are given. I don't take up the problem sets, but I may pull questions from the problem sets and use those as questions on your quiz. So that should sort of hopefully drive you to actually do the assignments to make sure that you actually have a proper answer. And so now what we'll do, we'll take a brief walk through Blackboard. So this is the home page for the Blackboard class, and the place I'm at has slow internet. So as you can see, I have most of the important tabs for today open in Blackboard. So the home page will always get you back to the home page. The mastering content is for the mastering biology stuff. Administrative is for things like your syllabus, and then we have each of the units. So I'll sort of walk through this. First, dealing with the content. Again, this is where most of your mastering biology related stuff is. An important piece in here, though, is your e-text. So click here, and the e-version of your textbook will open. And so this is open to chapter 2. The first time through, you will probably have to do something related to the rights and all that stuff that you normally see when you're dealing with new copyright content. Go ahead and approve that, and you'll be good. The study arena gives you extra materials that you can use to help additional quizzes and so forth. And it can also take you directly to your e-text. When we do have mastering biology assignments, they will be here. So I do have one that's ready for you guys already that will be due next Tuesday. So again, we'll talk more about this one on Thursday, but if you want to go in and take a look at it, you are welcome to play around with that. You guys did a lot of mastering biology work in your biology 120, 121, and 205 sequence, so you guys should be fairly familiar with how mastering biology works. You're not interested in you doing this introduction for non-major if you want to play it, so it's not available. But this first assignment is for you to do. We can address questions related to that when you guys can actually talk to me on Thursday. The next important thing is your administrative section. So this lecture PowerPoint is there. The overview, the review of Biology 120 that I showed you with the objectives that I want you to know related to bonding, etc., is there. Your syllabus is there. The the course schedule is there as well. So we will step into that for a moment. Maybe. So here's the course schedule with the topics that we'll discuss and when your exams are. And we hold pretty tight to the schedule. You should be aware of how we're doing this, because I stick pretty tight to this throughout the semester. Even days that I may have to be out of class, there's enough structure in terms of what we have on Blackboard that you can still accomplish everything that we need to accomplish from the class on that class date. All right. Also, your first quiz, your first online quiz is there. So there is a quiz on the Biology 120 review material. It's one of the online quizzes, so it's a closed book, closed note quiz. Again, there are 10 questions each time. You have 10 minutes to complete the whole thing. Once you start, you can't stop the clock, so the 10 minutes are going to count up once you start. It's available until noon on the 23rd. And you can take this, again, the quiz, unlimited amount of times until the deadline. Your grade is the average of all valid attempts. Standard unit looks more like this. And so this is the biological chemistry one. So you have lectures that are up there. You have the important figures from your textbook identified, and all of the figures in the PDF if you want them, so you can mark them up. Supplemental reading. So this is from those 3 textbooks that I mentioned before, links to each of the sections. So if you want to see a different way of looking at it, and there's usually going to be some sort of problem set for you guys to take a look at as well. So there are in the lectures 2 different types of lectures. So one is overview lectures. So this would be back at the level of potentially your 120 class to make sure that you have the background foundation. In this case, there are multiple because it's broken down. Most of these are only about 5 to 10 minutes long, and so it's going to take multiple to get all of the material in place. More importantly, though, are the ones from the Fundamentals of Biochemistry series. So these are at the level that you need to know this stuff to successfully finish this class. So again, overview, more 120 level, background, get you somewhat up to snuff if you are behind. Fundamentals are really where you need to be. My recommendation as you deal with this is going to be watch your overview videos, take notes on those, take notes on that, sort of reconcile those notes to help find you the important stuff. And then once you're ready, go through and watch the Fundamentals of Biochemistry lectures and take your notes on that. And again, reconcile your notes to help you to identify what the most important pieces are. One last thing that's here on Blackboard is your discussion. So this is where, if you have questions for me, these are actually where you put them. So you have questions about the content itself. You can post them here where it says post your content question here. So what you could do is you could go in and you can create a thread. And note here what I say, if you have questions about course content, feel free to post them here. In order to receive a response, you have to post a suggested answer for your question. That is what you believe is the answer. Now make sure that before you post that you look at the questions that are already there to ensure the question is already been answered. So one question may be, what is the difference between a prokaryote and a eukaryote? And again, what you have to do is start with you give me what you think your answer is. A prokaryote is generally defined as an organism without membrane-bound organelles, while eukaryotes have membrane-bound organelles. If you're uncomfortable with putting your name attached to these, you can post as anonymous. But once you're done, submit. I get an email saying there's a question there, and then I'll go in and address your question at whatever time I get to it, usually within 24 hours. If you have questions about assignments and they cannot be found in the course syllabus or course announcements, then you can post here. Please provide a clear name, not just my question, and review the items that are already posted to ensure that your question has not already been answered. And the final section here is a talk amongst yourselves section. This area is for you guys to use. You can use it to arrange study groups, share resources, exchange notes, whatever. I don't spend a lot of time in that. However, I will go in and monitor for tone occasionally, but I do try to leave that space to you guys. So, additional note in terms of course grading, that's your final project. So I generally come back to this about a quarter of the way through the course, but I give a cancer-related topic to different groups, and you guys work as a group on this final paper. It's a 3,000-word paper and a 20-minute presentation. The 3,000 word is the minimum. If you do not reach 3,000 words, then your paper was not done and you receive a grade of 0. If your presentation was not 20 minutes, then you did not complete your assignment and you will receive a grade of 0. This final project does require reading and interpretation of primary literature, and that's a big reason why the course is going to be designed the way it is, with lecture occurring outside of the classroom, plus for some other reasons that we'll continue to discuss as I go through this. Be able to give time in class for you guys to learn how to read primary literature. So you got to get your background information, your lectures, your textbook reading outside of class, so we can spend time in the class doing more difficult things. Now, one thing that we do a lot with this class is concept of value knowing. So repetitive recall / repetitive retrieval practice. And so what the has shown us, what other scientists have shown us, is that retrieval practice on previously studied material makes the material more likely to be remembered than simply studied. And in fact, newer evidence actually suggests that retrieval practice may not just help you learn old stuff, but may actually help you to learn new stuff as it is presented to you as well. So one of the foundational papers on this was by Roediger and Karpicki. And this paper was Test Enhanced Learning, Taking Memory Tests Improves Long-Term Retention. And what their aims were to investigate the testing effect. And so again, that testing effect is that repeated recall under educationally relevant conditions, and to determine whether testing facilitates learning beyond the benefits of just restudying the material. Of studies that they did within this paper. And so the first one, what they did was they had students read a short passage that covered some general scientific topics, and then they either took a test on the material or studied again before taking a final test 5 minutes, 2 days, or a week later. And so what you see in black are the folks who just studied and then studied again, as opposed to those who studied and then took a test. And so what you see is maybe 5 minutes later, the study was more effective, but as we start looking at time away from the actual initial presentation of the material, those who did recall practice in the form of an additional test in between actually had improved long-term retention of the material. And a second form, what they had the students do was read a passage once for about 5 minutes, and then from there, things changed. So one group studied for 4 different 5-minute blocks. Another group studied for 3 different 5-minute blocks and then took a test. And the others studied once and then took 3 different recall tests. And what you see is that, especially for long-term retention, again, the use of the test led to significantly better retention of the material. Where does this fit in with our class? Well, it fits in with respect to the weekly quizzes. So this gives you a chance to use your to study the material. It also works with our in-class quizzes, and you use your mastering biology assignments as well. That gives a 3rd place for you to use this. Now, second piece, why do I not lecture in class? So this is what we call the learning pyramid. This helps to show how much material is retained depending on how it is presented / interacted with. And what you see is that the lecture is among the worst for average retention rate. And that doing things that involve active learning and discussion are actually far more effective. And so most folks actually still feel the need for lecture, and so that's fine. We present you with the lectures. But the other advantage of using the online lectures is that you can go back, pause, rewind as you need to make sure that you're getting, as opposed to having to sit in class and sort of try to keep pace. An additional study from 2014 actually also showed improvement in student success in classes when they had active learning as opposed to traditional lecture. And so what you see here is that were 21.8% when you had active learning as the way that the classes run versus 33.8% under traditional lecturing. That's a difference. It's a 55% increase in failure rates when you go through traditional lecturing. So we want you guys to be as successful as possible. So we do our best to avoid doing that does not necessarily lead to your success. So what different types of active learning are there in this class? They range from you guys working out outlines to making concept maps to working on problem sets and case studies to actually presenting the material, again, discussion of both primary and secondary literature, as well as your mastering biology assignments. Some folks may not be familiar with what a concept map is, so this is a sample concept map where basically what you do is you take major concepts from a unit and use those with connecting terms to sort of map out a structured way of actually taking a look at the material. So in this case, this is one on the biological chemistry, starting with the subatomic particles and using those to build all the way up to our major macro molecules, our proteins, our carbohydrates, our lipids, and our nucleotides, which are going to make up our nucleic acids. A quick note here on incompletes. In general, I am not likely to ever get one. From the student handbook, what qualifies for an I is a student who is otherwise passing, has for a good reason failed to complete all requirements. If you had a grade or 0 above and you had to go away because you may have had a severe injury or severe sickness and it caused you to miss the rest of the semester, then we could deal with giving you an I. But just because you weren't passing and need some extra time in the course, I don't give I's for that. And again, all I's must be removed within 1 year or will be turned into an F. Note that is within 1 year. We can, as we negotiate the terms of your I, in the rare case that I do give one, actually give a deadline that is less than a year for removal of. All right, so what are the things that you guys need to do before we have class on Thursday? So number 1 is go ahead and complete your review of Biology 120 that we found in the administrative section. And so that's that list of topics from Biology 120 that you need to go back through. You should take the Biology 120 review quiz at least once between now and then. If you have any questions on Thursday, go ahead and bring them to class with you or post them in the looking forward to a good year. And I will see you guys on Thursday.